Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.
Good morning, New York. That wakes them up every time. Okay, I want to welcome you to Experiencing Science Through Commercialization. This is the special section of the Interfex called Interfex Live. Our first session this morning, we're going to be talking about formulation and delivery of biologics and complex small molecules. I don't know about the rest of you. I've only spent most of my career doing that sort of thing, so I would assume there's going to be a significant amount of interest. So, Jeff is going to be Jeff Shumway from Millipore, Kevin Bittorf from Therapeutics, Magali Hickey from Alkermes, correct? I get that right? Hey, that means I can read. Very good. Okay, so let's get started. So let's, Jeff, why don't we define the complex small molecules, therapeutics? You know, let's level it out. What are we talking about?
Sure, and you know, it's a loaded question, of course, right? When we start,
that's the way we do that.
When we start thinking about small molecules, you think of all the classics that have been developed, but then you start thinking about the pipeline that we're seeing with classic pharmaceutical development, where the molecules are getting so large and so complex, they're breaking into these kind of funny named characters like brick dust or grease balls, and you start looking at the complexity of these molecules, and it's almost to the point of getting to small biologics, where you think of peptides that are synthesized now rather than produced by biologic systems. So we're looking at a very gray area where these molecules are getting so complex that they're starting to bridge the gap between the structures of classic small molecules and more fundamental biologics.
So while we're on the subject, so let's like biologics over the last 5 years. I mean, how does that, you know, what trend have you seen, Jeff?
So yeah, I mean, a lot of with the biologic development, there's two things that we've noticed. One is the fact that in terms of compliance, we're seeing a trend toward higher and higher concentrations, and a lot of that is based on patient compliance, self-administration, subcutaneous administration routes. And as you bring these biologics into closer and closer contact with one another, you start seeing issues such as aggregation and even insolubility and increases in viscosity. All of these challenges need to be addressed as we get to these more complex dosage forms. But the other aspect that we're noticing is a lot of modified residues that are part of these biologic systems to enable click chemistries, whether it be for vaccine development or antibody drug conjugates and biologics of that type. We're seeing more and more complex biologics that typically would be normally soluble and bioavailable, but because of the complexity and the modifications, we're starting to see a trend more toward the challenges of small molecules.
So Magali, let me just ask this question. I mean, we were talking about trends and where it's going and complexities. So in my experience, like Novartis, it would depend on what phase we want to start something at. So why don't we talk about, you know, what factors are we going to roll back to phase 0? We're going to jump in at phase 2. So why don't we comment on that? I mean, that kind of sets the tone as well.
Yeah, so that's actually a great question with respect to doing pilot studies or phase 0 studies. These are things we talk quite a lot at my company, especially because we develop long-acting injectable products. And whether it's a biologic or that type of parenteral formulation, there is an unmet need for both of those. So there's an unmet need for large molecules now that we understand the human genome. We know that patient compliance in a lot of the disease areas that we serve is very difficult, you know, whether it's mental health, schizophrenia, opiate abuse. So you want to have the flexibility of giving someone a long-acting injection which is going to protect them. However, that brings on all kinds of challenges for how do you formulate these compounds. And as many of you probably know, there's only a limited set of excipients that are amenable for parenteral formulation products. So when you see half of your pipeline be geared down biologics, if we look at what's been approved recently, and half of it is small molecule, there's a great little marriage of, okay, for solid dosage forms, I can use these types of excipients. They've been used by the oral route of administration. And one thing that we would like to advocate for is have the courage to take some of these excipients which may improve the properties of a parenteral product, but they'll be considered a novel excipient for that route of administration, but then qualify it and use it if it's in the best interest of the product and the patient you're trying to support.
You know, I've done a lot of this in the past, and one of the approaches we use, at least in the, I had worked at Novartis for many years, is that if it ain't broke, don't fix it. In other words, we stayed simple, we stayed, if this worked, we hammered it in every product to make every product work the same way. So when do you innovate and when do you stay the course? I mean, that's what I'm...
Yeah, so you need to innovate at the very, very beginning. When you're establishing your research target product profile and your product profile, you have to make a decision. Am I going to go down the route of a fit-for-purpose formulation and I'm going to pay later instead of pay now? I think if I were developing a compound for oral administration that's not brick dust, I would say, you know, if it's highly soluble and it's a novel compound, go with powder in a capsule all the way to phase 2B. However, if you're developing a long-acting injection, you need to start innovating really early. And if you need the drug to remain in a depot for 1 month, 2 months, 6 months, you have to be willing to consider not just a different type of formulation, but also a device that can allow that to be delivered.
Can I comment on your phase powder and capsule to phase 2B? You know, it's always a nice idea to go powder and capsule to phase 2B, but it depends on what disease area you're looking at. Absolutely. If you think you're going to get fast tracked, you actually probably want to do a formulation in a proper formulation in phase 1, so you can actually get fast tracked and hopefully approved in phase 2B. Yeah. But
no, I agree totally. And to answer the direct question that you asked, phase 0 pilot studies, FDA is sort of looking for people to do more of that because getting the formulation right is something that's critically important. And if you can have early insight into that, depending on what the mechanism of release is, considering following the guidance for phase 0 pilot studies and talking to FDA about it may actually shorten your drug development life cycle pretty substantially in the end, and you learn a lot more about the compound.
That goes without saying. I always tell guys I work with, if the guy's selling a capsule, the odds are he's selling his clinical service form. What we did was we made sure that our clinical service form, if it worked, we didn't change it. We went to market. That's what post-market changes are for. Yeah. The answer is to get to market and to make it available. So, you know, just that's kind of where we're going with this. The other question I had was, Jeff, so based on what we just talked about, where the trends of formulation going? I mean, I know how we were talking with clinical service. Where are we going now? What are the trends like?
So, you know, it's largely with the small molecules, 90% of the pipeline is either difficultly soluble or difficultly absorbable. And in terms of the formulation strategies, you know, the golden arrow is to be able to administer oral, right? And we're seeing a lot of efforts put into dosage forms for oral availability of biologics, for example, which is no easy task because the body's designed to digest proteins, right? And so means for protecting those proteins in oral dosage form, that's kind of like the holy grail of formulation these days. Now, that being said, the complexity that we talked about earlier is really the key, because the 90% of the pipeline right now for small molecules is you need something to enhance the solubility without reaching toxic limits, because a lot of these molecules do have quite low toxic thresholds. And then as Magali was brought up, you know, these sustained release modalities are becoming more and more important when you get into molecules of that type.
Right. So Kevin, so how much emphasis, like at Trek, do you place on formulation development in the early stages? And how much do you put out there right up front?
Again, it depends on the molecule. If it's an insoluble molecule, which most of ours are, we put a lot of emphasis on formulation development earlier. We look at multiple different ways. And we even go back to the chemists as well, saying, hey, can we do something with this molecule? It's not soluble. Can we do something to change the molecule around to make it more soluble? Often you don't get a lot of success with that, but it's at least an option to look at. And then from there, we'll look at, we'll do a complete formulation screen, spray drying, hot melt extrusion, other soluble excipients. Me personally, I try to avoid soft gels because I go into areas that Middle East, Asia, where it's really not amenable to a soft gel type formulation. And I try to find a good contract manufacturer, contract development work, because I'm a 100% virtual company to work with. And I've had a few successes with a few, Juniper Pharma Services being one of the companies I've worked with and had a lot of success with. But I put a lot of emphasis early, because if you can't get it in, it's not going to work.
Yep. Kevin, earlier we talked about human factor engineering. You want to just comment on that a little bit?
No, human factor engineering, that's interesting. It's I look at it a little differently. Is like, how does the molecule go into the body, and how do you get it to absorb in the right spot in the intestine? And I'm working with a compound right now with another company. Is we're having an issue with it getting a little too. It degrades in the intestine very fast. It's like, how can we get it to slowly release and have it properly go into the intestine for absorbance into the body? Because right now it's such a high dosage form for the high dosage form for it. It's because of the degradation. We're really not that much is getting absorbed. So we're looking at right now engineering that product to have better absorbance.
All right. So I'm throwing this out to the three of you. Based on that, I mean, is there a preferred formulation or delivery method from a compliance, cost, or a safety? In other words, what you go to, what's it? This is my, this is my 8 iron. This is my go-to. So we'll start with Jeff.
Yeah.
Well, I mean, one of the most tolerable administration routes is oral administration, right? So if you have the appropriately qualified system of excipients and understand the physical chemical interaction of the API with the digestive tract and where you want to target it, then that obviously is one of the most tolerable administration routes. As you start getting into parenteral administration, then you have more complexities that you have to deal with, and compliance becomes an issue. And from a regulatory perspective, it's a larger hurdle to cross.
Magali?
Yeah, and I think I'm going to touch upon the point with respect to human factors. So my approach to formulation development, if the molecule is to be taken by oral absorption, by the oral route, and we've determined that absorption is not what's impacting the release, then you have a lot more at your discretion. If there's very high first pass metabolism, there is no formulation that's going to save you. You might have to consider changing the route of administration. So that's one way to think of it. I'm not a big fan of sort of the pharmaceuticals manual for the types of formulations. I really think it's critical to understand at the very fundamental molecular and chemical level why these inactive ingredients are in my formulation, and do I really need them? So one of the things that you see all the time is, we need colloidal silica, we need colloidal silica. And when I ask, why do you need colloidal silica for this particular formulation, I rarely get the answer that I want to hear, so we say that's coming out. With human factors engineering, so what that really is at its core, medical errors are the third leading cause of death in the United States. Of those, medication error, 30% of those, are attributed to your packaging and your labeling. So anything you can do from a formulation development, if you're developing a sublingual tablet, you better go do a study where you watch people put it under their tongue. And if they followed the packaging that you developed and they swallowed it, you have a real problem, especially if one of those components is not absorbed when you swallow it. It really needs to be under your tongue. So I don't think there's a one size fits all. I don't think there's a technology per se that can help with that. The most critical thing is think about the molecule, understand it, determine based on that molecule and the patient who's going to receive it, what's the best approach that you take forward.
So Kevin, let me jump in here in 1 second. So what I'm hearing is, the excipients, if I'm going to change the route of administration, okay, let's just say I got a novel excipient that I need. So I'm going to go, what's my path? I want to get this thing approved. So what have you seen? Sorry for the change up.
No, yeah, no problem. And I do want to comment on just excipients in general going into pharmaceutical products. For 2.5 years, I worked for a generic company, and I was baffled on what excipients went into this product, and I was forced to match the excipients because that's what patients look like when they compare drugs or comparing the name brand to the generic brand. They want to see the exact same thing. It's the I would say uneducated people that aren't skilled in formulation. They don't realize what these excipients do or don't do, so they just want them to be the same, especially for an over-the-counter drug. To get excipients approved, you're starting that in phase 1. You're working with the agency. You're doing a full tox program on your excipient, essentially, to prove it's safe and hopefully get it on the GRAS list. It's interesting working with excipient vendors. They don't necessarily want to do it. It would be nice if they did it themselves, but it's not cheap to do. But if you really need it, you have to go out and do the complete tox profile. It's almost like qualifying your API.
Well, that goes without saying.
Yeah.
Osmotics wouldn't be here unless somebody went about getting screwdriver handled plastic approved for human use. So,
Yeah.
Perspective. Yeah. But we're going to segue into that. So, Jeff, why don't we talk a little bit, because we keep talking about biologics and complex small molecules. So what are the differences in the formulation approach? I'm going to touch on that and we'll swing back.
Sure. So when we think about biologics, one of the discussion topics that we're hearing more and more of as we interact with more and more customers, especially ones moving toward continuous manufacturing approaches, is that with a biologic, it's been classically defined that we have a purification phase, and then probably when we reach our final UFDF, ultrafiltration step, that's where formulation begins, and there may be some compounding that goes on after that. But what we're seeing more of is, if you have a final formulation, a lot of the effort that's put in there for biologics is to improve the stability of that formulation, to keep the protein happy. A lot of those excipients and the chemical technologies that can be applied at the very end, if they were put into that purification process earlier, now you're stabilizing the protein through the process so that in your final dosage form there's less of a hurdle at the very end. So you can think of it as a continuous formulation approach. And that can lead to implications like selectivity of chromatography resins, because if you know certain buffer systems work better with your protein, you can select those buffer systems to help determine which chrome product you may use as part of the purification. Now with classic small molecule API formulations, you're making an API and then formulating it. And the bulk of the processing is really that formulation process where we talk about heat melt extrusion, spray drying, and all sorts of other excipient technologies we can put in there. And if I could just jump back to the comment around novel excipients and the chemist's comment about vendors. Coming from the vendor side, it's very, very difficult for us as a supplier to go out on our own and create a novel excipient. It would take 10 years of development, maybe 10 years beyond that before somebody actually adopted it. And nobody wants to be the first, they all want to be the first second, right, in terms of those novel excipients. So what we need too is better partnership between pharmaceutical manufacturers and excipient suppliers.
I've always found that being a fast follower was much more profitable for a company in the long stretch, but that's just me.
Yeah, and
just comment here. So, where we're at is, remember, we were dealing with biologics from in the traditional sense. If you talk to most people, we make the drug and we formulate it. And then they came into fill finish. Mm-hmm. Now we're saying, okay, let's load it up on the other end so that when we come off at the end, it's pretty much ready to go. So Magali, is that kind of where you see this going now? Or what's your comment on that?
So I think for biologics, and this is true for the majority of parenteral drug products, a formulation change for a biologic or for a parenteral product is very different than a formulation change for a solid dosage form. You have a lot more pathways there. So I think for biologics and for injectable products, you really want to spend a lot more time up front actually developing that formulation. And you made the point about novel excipients. We've had to do that. We took a product that an excipient that was approved for oral administration. We found that it was the only excipient that would allow us for an injectable to be in a pre-filled syringe. right, to flocculate as a formulation and be able to be delivered that way. We worked with the vendor of that. We did all the tox qualification and we gave it to them, and now they can use it for any future company that would like to use it. So there's a little bit of a scientific public service that you should do. Your strategy around qualifying novel excipients, Pfizer with HPMS AS did it with BASF, and we're all benefiting from that. We've done it with a particular excipient that we've now given. We're already aware of a couple of other products that are now doing that. So it is a challenge. You should never really go there first. But I caution against, I'm just looking at the GRAS list, or I'm just looking at what's on the approved FDA list. Do your research, see what products these are approved in an oral, and then have conversation with the regulatory authorities on what type of package you need to produce. Every vendor we've ever worked with is actually willing to do that with us and not carry the liability of what the tox package may insinuate.
Well, let me ask this question from a supplier and a user standpoint. This is directed at Kevin and Jeff. So in the formulation and drug delivery technologies, what is your perspective on novel excipients or drug delivery methodologies? I just want to get that sense. You supplier and user. What is your sense on this?
From a user standpoint, I work with a lot of my suppliers and vendors to look at formulation development holistically. So first of all, I always push, do we need this novel excipient? Yeah, and it's like, there's the cool factor associated with it. I have scientists that work for me and go, oh, this is just the next cool thing, let's do it. And then you have to push back pretty hard. It's like, why can't a basic excipient do it? And really put through your scientific studies and work with your vendors for the scientific studies or the device that you're trying to use and deliver. I've done a lot of formulation changes as well, looking at it was a novel formulation, but if you looked at it from a traditional sense, you could have used a different set of excipients and had more of a traditional tablet formulation. And that ends up being significantly cheaper to manufacture as well. So you really have to be careful when you're doing it, but I agree, when it's needed, you push forward and you get it done, and at the end of the day, it's for the final product as well.
Yeah, and from the supplier perspective, when it comes to novel technologies, we actually do at MilliporeSigma, we do a fair amount of work of not only custom API manufacture, but custom excipient manufacture, things like ionizable lipid species for delivery of some of these messenger RNA and interference RNA therapeutics. But another thing we strive to do, and we're doing a lot better at, is better understanding of the material science of the portfolio that we're presenting, so that we can present same technologies in different applications, and we can qualify them as such, and leveraging the fact that we do have toxicology data and other qualification attributes, then we can kind of open the door to novel uses of approved excipients.
Yeah, I do want to comment on material science. I think more and more pharmaceutical companies are getting very good at material science, and they're really looking at it compared to, say, 10, 15 years ago. They're really digging into the compactability profiles. A lot of people are using compaction simulators, looking at your excipient blends, looking at your API, just to see what's the compressibility of this, what's the minimum amount of excipients I can use, how fast can I manufacture this, will this work on a high-speed tablet press, and look at simulators to figure out the predictability of scale-up in itself, and really understand the material properties. And there's almost every pharma company now has a very large material science group.
Yep.
Yeah, and we've seen a big change, and I hope nobody in the audience is from the FDA, because I'm going to say something a little off color from their perspective. Excipients are functional, right? They all have a specific purpose. Magali indicated that earlier. If we can understand what functionally these excipients are doing in the formulation, what they're bringing, how they're interacting with the API and the environment to which they're being administered, we can do a lot better job at the formulation.
I agree with your statement. They are functional.
Absolutely. And I think FDA, cares about, is there going to be any pharmacology imparted by the excipient? But we all know that we're putting these things in there because they're gonna have a function, hopefully not on what the drug is supposed to do, but they absolutely matter. And if you're going to be the one that goes out there and does something that's different, and in FDA's eyes, unprecedented. So why are you the first one that's going to include this high amount of an excipient or a different type of excipient? If you don't have an answer to that, and a very good answer for that in terms of there were no other alternatives, then you won't win. Because FDA will always, rightly so, will say you don't know enough about this and tell me why you should be the first. FDA doesn't love the precedent, but that doesn't mean that they don't engage with you scientifically and propose a strategy that can make you successful.
It's always a trade-off. And a lot of times when you're going to throw a new technology, you're going to go to 505(b)(2) route.
That's right.
And invariably you're going to use resonation, you're going to use some other. The answer is that's when you get into the, well, I've never seen that much of that used before,
That's
novel
right.
or not. Okay, so yeah, it's something to consider. But I think the point there is that's why we're here. It's science through commercialization. Question I got for the three of you. So when you bring these novel processes in commercialization, which is why we're all here, right? So, what steps have you gone? We're talking about novel now, looks like it's going to work. So what's key about commercialization? We're going to leave the supplier to last. We're going to start with the users this time. So Kevin.
Novel processes. I worked at Vertex Pharmaceuticals for 8.5 years, and we always pushed on novel processes. I helped do the justification for the continuous manufacturing at Vertex Pharmaceuticals. And the first thing we looked at was, how will the FDA receive this? And we put a lot of effort into early communications with the FDA, early ideas on, can this actually blend powders properly? Looking at process analytical technology, and is the current equipment out there fast enough to measure what we need to measure at 20 to 50 kilograms an hour? So we put a lot of emphasis into analytical methods and communicating what our strengths and weaknesses of our analytical methods were directly to the FDA so they could comment. Even bringing them on site to look at kind of our pilot rig, our test rig, which before we bought everything else and had the full vision of developing continuous manufacturing. So it's a lot of working side by side.
So we're talking about analytical methods as well as process methods.
Yeah, they have to go hand in hand. If you're doing continuous processing, you need real-time measurement. So you have to know your material is well blended. You need to know once it's coming off that tablet press, the hardness, the thickness, the potency in real time, because you're just manufacturing 20 kilograms an hour, and you don't want to. You don't want to do it in a batch manner where you're just taking samples afterwards. It's like the whole idea of continuous manufacturing is taking that to a real-time release to get it to your patient faster. Almost a supply-on-demand technology, essentially, which eventually could be hooked up directly into packaging equipment, directly to the back of the truck, and directly to your distributor.
Magali, what's your take on
that? Yeah, I think what you're saying is dead on. And a lot of times what companies will do is they'll spend a lot of time on the material science, understanding the composition. Now I have my composition, which by the way is not independent of the process, so you need to do some of that work. But unfortunately, what a lot of companies do, they say, I have my composition and I've scaled it up to a pilot stage in batch mode. The first time I automated it is during validation of that product. And then the first time I do any PAT is actually when I'm in the middle of an investigation post-commercially. So what everyone should have the courage about is to demand the type of product that you want to develop is one where you can have eyes on it from beginning to end. In doing so, you will determine that for appropriate scale up, batch process may help me, but maybe I need to invest in continuous manufacturing because this is a very difficult process to make. So I think I agree with that 100%. But as you think about your facilities, you really have to think about the utilities. When can I bring automation during development, PAT, and really understanding the design space, because you will pay for it commercially if you don't pay attention to all that stuff.
You're up, Jeff.
And so when it comes to these novel approaches, right? We, as a supplier, we end up being a support factor for our business partners. If you think about something such as continuous manufacturing that Kevin was describing, it's on us as a supplier to understand where these products are going, and that relationship and that understanding is important, because if we as a supplier are only interacting with procurement, for example, and they're buying raw materials that are seen largely as commodities, When we talk about excipients, they're really not commodities. These are integral parts of the final formulation, and if they're being used in a continuous manufacturing process, and say, for example, particle size distribution is important to that process flow, if we can understand that as a supplier, we can help integrate that into the process. And not only that, for some of the novel delivery platforms that we engage business partners with, we can actually help write regulatory submissions because we understand the chemical material science of those excipients, and then we leave the formulation piece and the interaction with the API up to the real experts, which are the developers of the pharmaceutical themselves.
Okay, let's turn it around back again to the two development sides. So when is real-time release going to be a factor here, Kevin?
When is it is a factor. Real-time release, you wanted a factor. I've always looked at this for a supply and demand technology. So it keeps your warehouse space to a minimum, which saves you money. The ability to take it right from that process, right to the consumer. So for urgent indications like flu epidemic or pandemics would be a very good use of real-time release. And it shortens your supply chain. So it actually, there's a lot of potential savings in the shorter supply chain as well, and warehousing space.
Magali?
Yeah, I mean, and I see a lot of that stuff to really be about the quality of the product. There are so many benefits to have eyes on your process and to use continuous manufacturing. You're not going to fail as many batches. That doesn't mean that you're not. But the luxury of stopping and being able to go, rather than I stopped and I had to start all over, I think is a really important approach, and you should be doing it as early as possible. So develop your process with FBRM, develop your process with near IR, develop your process with CubeAD. If you need to sample and your tank doesn't have a place where you can retrieve that sample from, okay, if you're not going to do PAT, make sure you're talking to engineering and saying, these are the things I want my process to be able to do. This is what I want to be able to manage, because every process is designed to do exactly what you ask it. So we can't come 2 days later and say, I need to retrieve an in-process sample. That should happen at the very, very beginning. So you buy equipment that gives you that luxury, gives you that flexibility.
So we're talking about front loading a lot of the work here and cost, right? So yeah, I get that, all right, because you don't want anything sneaking up on you. That puts a lot of emphasis on you guys, Jeff. I mean, there's going to be specifications in the quality standard that before was never an issue. So the question really comes back to is, if I'm going to front load it, I'm going to put in this instrumentation in the end and want you guys to think about this. So what about the impact on cost of manufacturing? Okay, everybody's going to say, well, I'm going to do continuous because it's cheaper. Well, is it? I mean, when does that decision get made? So we're going to do it just the way we did before. We're going to start with Kevin, Magali, and then we'll get back to you, Jeff. So it gives you time to contradict everything they're saying. So, okay, so you're up.
I did the complete justification for why to go to continuous manufacturing from a financial standpoint, and that was the first thing I looked at. So the one thing that continuous manufacturing will definitely save you money on is on development. So you can stay at that small scale throughout your phase 1, phase 2, phase 3, and commercial scale. It's at 20 kilograms an hour. I always said if you want to make a 500 kilogram batch, well, you have to scale up at least once or twice to 500 kilograms, and that material is generally thrown out. When you're doing 20 kilograms an hour, you know, yeah, some of that's thrown out, but you're doing a complete design space. So you're using a lot less API in the development stage. And in development, your API is anywhere from $10,000 to $110,000 a kilo. So putting up to 500 kilos, even at 50% drug load, that's a lot of money from that aspect. So where it saves you money in production is there's a lot less waste. So you look at the waste from the API, because the startup and shutdown is a lot less as well, and you have a lot less material storage. But from a development standpoint, you save a significant amount of money.
Yeah, and you know, I echo everything that you say. At my company, when we're making decisions about going with a batch mode for something versus continuous, it may look on paper that going with the batch mode is going to be cheaper. So I'm not even going to comment on what it means in development. My answer is always, none of that cost will account for batches failing in commercial development and not being able to sell that product. that happens, the more complex the process is, the more susceptible you are to doing that. And that should be what the debate is about, not what is going to be my cost during development and sunk costs, right? You can't think about sunk costs. I invested in all this batch process. I don't really care. I care about you being able to reproducibly manufacture this drug, because for the disease areas that you might be in, so we work in opiate relapse, there is no such thing as I have a drug shortage right now because I don't have a strong enough process that doesn't fail. So I think if we bend the conversation that way, I think people will give themselves the permission to actually say this cost on its own in a vacuum is not the whole story. It's a part of it. And depending on how complex the product you're developing, it's a very different model.
Yeah, well, I was really happy to hear that it wasn't excipient costs that were one of those issues. But you know, it's interesting because what we're talking about here is kind of processing with the formulation in mind, right? Working our way through to build a robust process. And for us as a supplier, you know, let's just take a simple example like mannitol. If we know that mannitol is being used for direct compression at the end of one of these processes, we know that we can define certain quality attributes about the particle size distribution that make it very amenable to the process and blending with the API that's produced from that process. But if we take that same mannitol and say it's being used as a stabilizer in a biologic, now there are different quality parameters. So we have to qualify the materials, or rather the excipients, based on the application so that it fits better into that process stream.
So, manufacturing costs, excipient costs, key factor, yes or no?
Key factor in what?
Cost of products.
The
as an industry, we get hammered, right?
The price of products. No, manufacturing, the price of products is based on a different set of group. You know, obviously if your manufacturing costs are higher than your price of your product, that product's not going to market. That's when manufacturing costs become critical. At the end of the day, it helps the corporate margins, reducing your manufacturing costs. And pricing is determined by going to your payers and what they'll pay, because at that point when you're putting it to market, everything else is a sunk cost. It is expensive to take a drug to market, $1 billion to $2 billion, but manufacturing costs help the company save money so they can invest into more research.
Yeah, and again, I'll try to be a little bit provocative. My answer is no.
It's no.
Okay, but at the same time, just like we talked about novel excipients, I need to do this because it will make the product in the end better. I think one of the things that really squelches innovation, both on the technology front and on the formulation development front, is when we tell people very early on, you really have to have the cost of goods in mind. I'm not saying ignore it. However, we should be willing to push back, and we should be willing to say we need to develop the right process. It's a negotiation. And before you spend any money, have the negotiation debate. Don't just go without having that, because the minute you get on the train, you might not be able to come back. So yes, companies are concerned about sunk costs. What I have seen in our industry is usually the process that wins is the process that's a little bit further ahead. It's time. It's not what's better. And until we as an industry start to say, time is not everything, we understand what that does to the bottom line, but please send all your scientists to business school, because losing batches commercially and losing production commercially, it's not even worth the conversation.
What's your perspective?
So, you know, I was looking at some statistics, and you know, biologics obviously are, you know, kind of a hot topic right now. And if we look at the cost, if we exclude cell culture media, which is very customized, and we look at the cost of the chemical technologies that go into a purification process, it's less than 1%. Less than 1% is that raw material cost of either process chemistries or excipients at the end. When we look at small molecules, it's greater than that, right? Because we're talking about a solid dosage form that isn't predominantly water. But the reality is, most of the cost in these processes is the manufacturing and everything that goes into it, all of the inputs, whether it be qualification of the materials, whether it be analytics. And if we can do better as an organization of suppliers to provide that information up front, it's going to save money on the manufacturing cost, regardless of what the cost of the excipient is.
So, we put the time in up front and we run it continuously, we're in stasis. The answer is, good science says it's going to be good. Mm-hmm. Okay. But, this is something, and this is the last question, but the question's only, and we're going to talk about this again in our next session, which is going to be on continuous, but let's talk about turning it on and turning it off. I think there's a big misunderstanding here. Yeah. So we're going to start in the middle this time. So Magali, remember, management, remember all the stuff here, we're going to send our scientists to business school, but on the same token, management thinks I can turn this thing on and off like a light. So, comments on that?
Yeah. So that's an excellent point. A continuous process is not infinite. You are going to have to qualify the process running for X amount of time with these critical quality attributes. These are the outputs, and that I can do that in a manner that is controlled. I like to think of continuous manufacturing. So, for example, if I have a recrystallization where I need to set the form and the particle size of an in-process material, I know it's critically important. I know it's very difficult to scale. I can make that part of the process continuous. And then when I go to make the suspension formation, whether that needs to be continuous or not, or go from beginning to end, there is flexibility within that to develop the product the way you want to. But where I have found it to be most promising, if you will, is in solving technical challenges that you can't do as easily in batch mode. But yes, there is of course a beginning and an end, and you really have to validate and qualify the robustness of every part of your continuous process.
Kevin?
Yeah, it's not on and off because every product almost has a different process or a slightly different process. So if you have a continuous processing line, there's change parts you have to do. You have to clean between products. There's obviously the cleaning for a continuous process is quite a bit more complicated than a batch process as well. So, and just adjusting the equipment and making sure it's ready to go. So from an on-off, often your changeover time is anywhere from 5 to 15 days between products for a continuous process. So it's letting your management understand that and really having your management and supply chain plan ahead so you can do a longer run and make material for the year or for the half a year that's needed versus multiple small batches. And if you're doing that and your process allows batch processing actually might be cheaper.
That's right.
But you have to really take a look at what are you using the continuous processing for.
Yeah.
Is it to save time and money early on, which is a valid reason, or is it to save production time and money later on, which is another valid reason? And obviously there's your technical reasons that this is the only process it works on, and that's another valid reason to use it. So you really have to look at all the reasons why to go to continuous processing or continuous manufacturing.
Well, we're just about out of time, but I think you could start to see that the lines between the types, dosage form types of API, the types, they're starting to blur out. Continuous process, rather than being the scientific curiosity it was, it's all part of our decision now. We haven't even touched on scale up, scale out, multiple clones. This, again, we're going to do this in the next session. With that, I want to thank our panelists, Kevin, Magali, and Jeff. Thank you very much. That's it.
Thank you. That was good. That was fun.
Pharmaceutical Processing, in conjunction with Interfex 2018, presents Interfex Live.